Hello and welcome to another episode of Physiology with Shakti. Today we're going to be discussing about your neuromuscular junction. Today especially we're going to be talking about your skeletal muscle and the end plate that is being formed. So if we have to start off, we'll start right from the axon which is coming, and this axon basically comes from your spinal cord, from the motor neuron that is coming, and this at the end goes into temporal arborization. It splits up into multiple branches, and that is where you have the terminal button like structures which are developing. Now just draw a diagram of the muscle at subline, and around this usually we have the basal lamina which is present, and this basal lamina in turn has post-junctional folds which will be visible like this. Okay, now we're going to zoom into this further and take a look at the structure. So what you can see here is that terminal button. I'm zooming into the terminal button, and that is the presynaptic terminal, and beyond it is a postsynaptic terminal which has been specialized, and you're able to notice that they are thrown into junctional folds actually. There are clefts which are developing over there with the crest of it. So this is a specialized end of the post junction, and there are some specializations that are also happening in the pre-terminal. Okay, presynaptic terminal. So whatever neurotransmitter is coming, usually it is carried over through your microtubular structure, and there are vesicles in it. So that is certain type of neurotransmitters. So these vesicles appear like dense core. Okay, so usually the peptide hormones and all, and the neurotransmitters which are preformed and then transported down, that will be present in this dense vesicles, preformed also. Beyond that, we have another type of vesicle which is relatively clear vesicle. So in these clear vesicles, there are no preformed, but these vesicles reach inside the terminal end, and then things are transported inside. One example is that of your acetylcholine. So in acetylcholine vesicle that we're talking about, around 6,000 to 10,000 acetylcholine molecules are there in one vesicle. Now, if you look at your presynaptic terminal, there are certain areas of the presynaptic terminal over here, over here, over here. Okay, so what you have to understand is these are areas which are right opposite that post-junctional cleft. Wherever the folds are happening, just opposite that, these are certain areas which are specialized in the presynaptic terminal. And these specialized areas are what are called active zones. So there are certain active zones which are present. What is the peculiarity of this active zone? These vesicles containing neurotransmitters, they come down, they dock at this active zone. And how do they dock? They usually have a linear sort of arrangement, and they are in twos actually, rows of two. A linear arrangement is made like this. So three-dimensionally it just goes back and linearly in twos it is actually arranged. It will be there on this side also. Okay, so at the active zones, that is where the docking is taking place. In the presynaptic terminal, apart from these two, we also have a PQ type of calcium channel through which calcium can come in. And you also have to draw your mitochondria over here. Okay, so these are basic things that you need to draw in the presynaptic terminal. Okay, we're not done with this yet. We'll still come back to it. So now what is going to happen is these vesicles which have docked right now, Okay, so we'll go into understanding the concept behind the mechanism of docking and exocytosis. So the first thing that is going to happen is what we call docking. So what happens in docking? So we have the uh, vesicle containing the neurotransmitter, and on the opposite side the presynaptic terminal is what we are drawing over here as a membrane over here. Now in the vesicle there are a certain group of protein structures which are involved. These protein structures which are involved are, since it is present at the level of your vesicle, it is called V-snare. So these are V-snare proteins. Now the V-snare proteins, specifically in your neuromuscular junction, what the protein that is specifically called is synaptobrevin. Okay, you can take a look at this V. There's a brevin win for V snare and V for vesicle. You can remember it like that. On the other hand, at the level of the presynaptic terminal, you have another substance like this. Okay, this is what is called a T snare. T snare specifically in this area is called syntaxin. Okay, so T for and T for this. Now beyond this we also have another substance which is there. This is what we call SNAP 25. So this is a complex which is there. Okay, so this is how the docking is taking place. The vesicles are coming closer. That vesicle has a V snare. On the opposite side there is a T snare as well as SNAP 25. Also here there is another component which is there, which is called synaptomin. So this component is called synaptomin, which is present. So after docking, we're gonna go to the next stage of the step. The next stage of the step beyond this, we're gonna call it the priming phase. And priming is divided into two phases, phase 1 and phase 2. So in the priming phase, what is going to happen is this vesicle that we just saw, that vesicle is going to come closer and a ternary complex is going to form. So there is an interaction between the V snare and your T snare, resulting in a ternary complex. Okay, so thus because of this priming phase, there is going to be formation of a ternary complex. Okay, once this ternary complex is formed, the next phase of priming happens, which is called phase 2. So in phase 2, what is going to happen is, a stronger binding happens. Okay, further stronger binding happens of the SNAP proteins, and another cytosolic protein is gonna come. Cytosolic protein called complexin. So another cytosolic protein called complexin is going to come, which will attach itself to the snare complex. So what it essentially does is prevent the fusion and release of the neurotransmitters. Okay, so you can see a tighter complex being formed. So just for a diagrammatic representation, I'm just going to draw one structure over here. Okay, as if something is there to prevent the fusion of this neurotransmitter containing vesicle, and that is your complexin. Okay. The next thing that is going to happen is your entry of calcium. So this is where you have to understand the importance of this structure that we were talking about, which is called synaptomin. The structure called synaptomin is there, and this is where the calcium which is coming inside will come and bind to the synaptomin, resulting in this getting attached in the membrane structure, and your complexin is removed. Okay, so entry of calcium, the synaptomin is activated, resulting in this inhibiting your complexin, removing it, and thus there is gonna be a fusion. So that fusion was stopped by this complexin actually. Okay, so this will come down and there will be a fusion of this vesicle. Okay, now after fusion it's still not done. After fusion, this is when the step of disassembly is going to happen, and for this step of disassembly, two structures are involved, which is NSF and an alpha soluble NSF associated protein, which is called alpha SNAP. The main thing here is we have a alpha SNAP, and above that we have a NSF. So the whole point of NSF is to utilize ATP, resulting in hydrolysis, and with that energy that is formed, it breaks this ternary complex and brings it back to its original state. So while this is happening, two things will happen. One, the T-snare component, that will be ready for another round of this whole process. On the other hand, the V-snare component, which is there in the vesicle, that will undergo a sort of a recycling endocytosis. So we'll get endocytosis, will go back inside and go into the endosome where recycling can happen. Okay, and then it'll come back down as a vesicle again. Okay, so these are the various steps that are involved in your exocytosis of vesicle. So it's not that simple as you can imagine. Okay, so it's not simply like calcium comes in and immediately it goes into exocytosis. So you have so many things to explain under this. Okay, so now that we covered that part, we'll zoom in again. So this is where you have to understand that basal lamina we were talking about earlier. Inside the basal lamina, there are multiple structures like this. So it's filled with, collagen and proteins, proteoglycans. And apart from that, along with that is another component which is there, which is an enzyme. So which is called your acetylcholine esterase, which will be there throughout here and there and everywhere. Will be there throughout in this space. Okay, so that is basically your acetylcholine esterase. Now this has been released and it reaches the synapse. So this area between this is basically called your synaptic cleft. So this small area between that is called the synaptic cleft. Okay, it's going to traverse through the synaptic cleft and go to a receptor area. So remember how we were talking about the active zone in the presynaptic terminal? Just like that, there will be a particular area where you will see more of your receptors. So that is basically, this junctional folds which are there, the crest part of that junctional fold that you can see here. Okay, so this junctional fold, the crest part of that junctional fold, that is where you see more of these receptors. So what is this receptor called? This is a acetylcholine receptor, which is nicotinic type. So I'm going to represent it as a NM. Okay, so your nicotinic receptor, so it's a acetylcholine nicotinic receptor, that is of NM type, which is there in your skeletal muscle at the neuromuscular junction. And we have a N type. This N type is present in the ganglia. Okay, so this receptor that we are talking about, it has a pentameric structure. This receptor has one alpha over here, one alpha over here. This is an area where beta is going to be there, a gamma and a delta. Okay, so this is how the receptor structure is, which is pentameric in nature, and there will be two regions between this alpha and delta, and between this alpha and gamma, where acetylcholine can come and combine. And mind you, we need two acetylcholine over here. Two acetylcholine should be present to actually result in a open state of this receptor channel. So this receptor is also acting like a channel when activated. When acetylcholine is there, 2-acetylcholine is there, it becomes an open state. Okay, and in the open state, it is acting as a non-selective cation channel. In the open state, it is of course a cation channel. Negatively charged ion particles are not going through it, but it is non-selective, and because of that, there is influx of sodium, potassium, calcium. But studies have shown that the major role is played by your sodium and potassium over here. Okay, so sodium and potassium influx is going to happen. Because of this influx, that will generate a end plate potential. Okay, so this is a type of a graded potential. So it's localized. Okay, it does not go that far, it's not progressive. Okay, and it can be summated upon. So more of this happens, it keeps increasing. Ultimately, when it reaches a threshold, it goes into a action potential. So till that point, this non-selective cation channel opening will result in a end plate potential. Okay, now once this end plate potential reaches a particular threshold level, that is when another group of channels are opened up over here, here. Okay, so that opening up of these channels will result in sodium. So this is a type of voltage gated sodium channel. Okay, so end plate potential, once it reaches the threshold, it results in activation of voltage gated sodium channels. Okay, so we have a voltage gated sodium channel opening up, that will result in your action potential. How does this action potential look? Just like your normal spike potential. Okay, because this is a skeletal muscle and sodium is involved over here. Okay, so again, once you acetylcholine is binding to acetylcholine is binding, it becomes an open state where non-selective cation channels are moving in. That results in an end plate potential, and once it reaches a threshold, voltage gated sodium channels are activated, resulting in an action potential. So while experiments were done, one of the things that was discovered was, in random state, there was always, a minimum of zero point 0.4 to 0.5 millivolt of current sometimes passes through because of movement of ion through these channels. Okay, so what was understood is a particular minimum quanta is being released, the minimum that is possible, and anytime we need more, it is multiplications of that quanta that is being increased. Okay, so this particular concept is what we call a miniature end plate potential. If you hear a term miniature end plate potential, that is, basically what is released by one vesicle. That will result in one quanta, and that corresponds to around 0.4 to 0.5 millivolt of the current. Okay, so miniature end plate potential is talking about whatever electrical current will pass through these receptors when one vesicle is released. That one vesicle corresponds to one quanta. So if you want higher grade of activity, more vesicles are released, but it's always going to be in multiples of this particular unitary quantum. Okay, so that is the concept of what a miniature end plate potential is. So we understood what is the end plate potential, miniature end plate potential, how action potential is developing over here. Now once the action potential is developed, now what has to happen if it continues staying there? We don't want a continued depolarization. We want the acetylcholine to go out. Okay, so earlier we were talking about clear and dense vesicles. So this clear vesicles that we're talking about, that is an example of your acetylcholine. Okay, so imagine there is a clear vesicle over here. How do you get acetylcholine? So one of the things this acetylcholine esterase does, that acetylcholine esterase takes the acetylcholine which is over here, splits it up, and that can be reabsorbed inside. So what is happening? You have your choline component, we have the acetyl-CoA component. So we have acetyl-CoA and choline. So these together form the acetylcholine. Acetylcholine esterase, what does it do at the level of the receptor? It splits this and helps it take it back eventually. Now once it is taken back, this will together again form your acetylcholine. Now how does acetylcholine go into the vesicle? So acetylcholine, when it is going into the vesicle, before that there is a step where H plus is put inside actually. Okay, so acidification of the vesicle is one thing that happens. Why there is need for that? Let me explain to you. Because there is another exchanger over here which takes H plus outside and in turn takes the acetylcholine inside. Okay, so this V type H plus pump is important in order to maintain the gradient for the exchanger to take acetylcholine back inside. Okay, so that is how the acetylcholine esterase works, then how choline and acetyl-CoA together forming acetylcholine and is taken back inside. And this is one of the reasons why pH is something that can affect your, not only here, all sort of synaptic transmission in general. Okay. So this is where you have to understand the clinical relevance of whatever we talked so far. So these SNAP proteins, there are certain toxins which can inhibit them. So we have the tetanus toxin that inhibits your synaptogrelin. Okay. Then we have botulinum toxin. Botulinum toxin, there is, different types which are there. We have A, B, C, D, E, F, G. Out of this, your A and E, that inhibits your SNAP25. Okay, and a particular type of C, like the C1, is inhibited. Okay, the C1 inhibits your T-SNAP, and the rest of it inhibits the V-SNAP. So B, D, F, G inhibits the V-SNAP. So in different regards, that causes a problem with the exocytosis process over here. Apart from this, when autoimmunity is developing and the autoimmune antibodies go and attack your nicotinic muscle type acetylcholine receptors, that is where you get your myasthenia gravis. And instead, if it goes and attacks the P by Q calcium channels presynaptically, that is where you get your Lambert-Eaton syndrome.